Let's get everything up and running. Quick show of the pups. There's one, and there's number two. Hopefully tired out after their walk this morning. So they are going to behave. Okay, so let's get to it. So where we ended up last time is really talking about all the different layers and levels at which gene expression can be controlled in eukaryotes. And so we're going to pretty much just start at the top and work down, right? And go through all of these one by one. And in some ways as well, we start off with the broadest scale, like the biggest picture part of it, right? And so remember I was talking about kind of developmental programs and stuff like that, right? Where, you know, you can have a whole bunch of genes being expressed that are involved in the same process. Well, quite often those are actually grouped together in the same region of the chromosome, right? So you can change the chromatin state of that region and shut down the whole program at once without worrying about each individual gene, right? And so really, when we talk about chromatin, we're talking about epigenetics, right? So you remember chromatin, let's just get this up. Chromatin is DNA and histones, right? And so DNA in eukaryotes doesn't exist unless it's being synthesized in any other form, right? So when we talk about DNA, or actually we're talking about chromatin, right? That's the reason for the proteinase K digest that you did in lab when you did your Drosophila DNA extraction. It's not to get rid of the proteins in the cell, it's to get rid of the proteins attached to the DNA. And that chromatin, its properties can be changed without affecting the sequence of DNA. Okay, and these properties are heritable. That's the crazy thing. And in fact, so actually I'm going to talk kind of big picture first, right, before getting into the details. Right, if you think about us in our environment, right? So imagine you kind of cast your mind back to, you know, when we were cavemen running around pushing woolly mammoths off cliffs and stuff, right? So the environment is constantly changing, right? The whole ice age thing and, you know, famines, food abundance, et cetera, right? Droughts, various changes in the climate and the environment. So our DNA doesn't change that fast, right? Changes by random mutation, right? And you have no say over, really, as an organism, what those changes are. They just happen randomly. Hopefully they benefit you and get passed off onto your offspring, and, you know, maybe they don't. However, you need to, like, for the survival of your offspring to be optimal, not only do you want to randomly mix together all these alleles to create potentially better combinations, but you also want to kind of give your offspring a bit of a heads up, essentially, before they're born. And so epigenetics is really a way of speaking forward to the next generation in many ways, right? So we're going to be talking about epigenetics in the context of, you know, changes within the organism itself, right? But also as a heritable property. This is a way of kind of speaking forward, essentially, right? So if you're currently in a famine condition, severely nutrient limited, it benefits your reproductive success if you just go like, oi, life sucks out here to your kids before they're born, right? So they're prepared organismally, right, in terms of metabolism and the like, before they're even born. And that persists for, typically about 3 generations before the effect peters out. Because essentially it gets diluted every time you reproduce, right, for each generation. And so just to blow the minds, at least of the ladies present, right, for all of you ladies, you made all of your oocytes, what will become or may never become your eggs as you ovulate every month, when you're inside your mother. Okay, so the oocyte that made you, right, because obviously it's an oocyte, or it was an egg essentially, but an oocyte that changes into an egg. The oocyte that made you was being made by your mother while she was inside your grandmother. So you can have 3, for the females at least, you can have 3 generations in one body, right? So you can imprint information onto those daughters and granddaughters to prepare them for the environment that they're about to encounter. And obviously you don't want that to last forever, right? Because, environment, changes essentially. And so that effect dissipates over generational time. They actually studied this in relation to a famine during World War II in, is it Denmark? No, it was in Holland, called the Hunger Winter. And they've actually detected changes in the grandchildren alive now whose parents were in utero during that famine in World War II, which is absolutely crazy. So anyway, that's kind of the big scale stuff. Going down, we're talking about changing large patches of the genome, and even further, we can go down to changing small patches of chromosome, right? And these modifications are made in a bunch of ways. They're made in terms of like the physical state of the chromatin, right? These are changed by chromatin remodeling complexes, which use ATP. And then we're going to talk about, in actually quite a deal of depth, actually, about the chemical modifications you can make to the histones and to DNA, right? And these are astonishingly complex. So much so that I was doing some reading on it yesterday and I had to stop because my head hurt. Just lay down, dark room, eyes shut for a little while, right? It's crazy, crazy, crazy complicated, right? And so this epigenetic code, right, that accumulation of all of those different changes is just astonishingly complex. Actually, another fun fact, while we're on the subject, identical twins have exactly the same DNA, right? That's why they're identical. And when they're born, particularly if their parents like dressing them the same, you can't tell them apart, right? They look identical. That's why they're called identical twins. However, as those twins get older, you'll start to see subtle differences emerge. That's not because their DNA is changing or differs, right? DNA doesn't change that quickly. It's because of epigenetic modifications. And so based on the experiences, the various interactions with the environment that those 2 twins have, they will become subtly different over time. So by age about 50 or so, you can reliably tell identical twins apart, even if dressed and haircut and whatever the same. which I think is pretty cool. But then again, you know, genetics is cool. What can you do, right? So anyway, that's the first one, and then we'll spend a whole bunch of time on the second and third kind of modification, right? And so when we talk about chromatin remodeling complexes, we're really talking about how the DNA is associated with the histone proteins, or really whether those histone proteins are there at all. And so, like, the overall big point to get, right, the thing that you really must get to understand all of this, it's all chromatin and chromatin state is all about access to DNA. And I'll write that down because it's important. And this is, oh, actually, can anybody remember the two different general states of chromatin? It's something chromatin and something chromatin.
Heterochromatin and euchromatin?
That's 100% correct. Can you remember which is which, open versus closed? Active versus inactive?
Euchromatin is open and heterochromatin is closed.
That's awesome. Thank you, whoever that mystery voice was. Sorry, I can't actually see who's speaking. Yes, exactly. And you know the way that I remember it, this is kind of, it's a little bit of an off-color joke, to be honest. So take it as you will. But back when the whole Syrian refugee crisis was happening, you know, EU was being very open and welcoming. Obviously, it's not anymore. It's like, hey, go away. Don't want you lot, right? But I always used to think, oh, the EU is open, right? EU, euchromatin, open chromatin. Anyway, it's not particularly great, I admit. It's not my best one, but it will do. So use that if that helps. Right, so chromatin state, and that's like hetero, oops. And that's closed, and there's kind of varying degrees of that too, right? So it's a very dynamic property versus euchromatin, which is open. And when we mean closed and, sure, what was the point I was making here? Oh, right, got it. When we mean closed, we mean not accessible. Open, accessible. If it's open, it's available for transcription. And the reason why accessibility of DNA matters is because in eukaryotes, gene expression is off by default. And so you need proteins to come in to turn on gene expression. If those proteins cannot access the DNA, that gene will not be transcribed. As TBP, activator, RNA pol, etc., to bind. So if you think about this, this is very much like the higher order stuff, because even if all those proteins are present, you have all of the transcription factors and activators and yada, yada, whatever you need for transcription to occur, and the DNA is closed, is inaccessible, you don't get transcription, right? So this is like the gatekeeper in a sense. And this is shown really nicely here because, you know, you have DNA that's in the default state, is tightly wound around these histone proteins. Well, we've got a nice picture of histone proteins. All right, and they look like that. It's an octamer, and they have these tails kind of hanging out the side, right? So in the default state, DNA is tightly wound around that core particle, right? Those 8 histone proteins. And there's a bit of DNA between those nucleosome core particles, about 50 base pairs or so. However, if the DNA is actually wrapped around that particle, that DNA is not going to be available for transcription or binding, more importantly. Okay, hopefully that will make sense. If you got any questions, just, pipe up and give me a shout.
So you said that when it's binded around the histone, it's not available for like transcription. What about like the TAP binding site? If that protein binds there, does it make that section of DNA? Or what are those for?
So TAP binding site is for the transcriptional activator protein. I'll just write that out. Give me a second. And so proteins need that bind DNA, the DNA binding proteins. They recognize a sequence, right? A particular sequence of bases. That's what they're looking for essentially. And they are kind of, it's not just like a 2 bits of paper lying on top of each other. Those proteins actually fold around the DNA. And so if that DNA is very kind of closely packed to something, those proteins cannot properly kind of glom on to that sequence. And so these sequences here, this is TBP is the Tadalbox binding protein. Right, so the Tadalbox binding protein brings in RNA polymerase. The TAP, transcriptional activator protein, either brings in TBP and a whole bunch of other stuff, or it actually activates the complex, right? So if those sequences are not available, even if one of them is not available, then the whole transcriptional complex cannot assemble on that promoter. Does that make sense?
Yes, thank you.
Yeah. So it's all about accessibility, right? And you'll see later that this DNA can still be wound around the histones, but if it's very loosely wound, right, kind of, just like baggy trousers kind of deal, it is more accessible. Whereas if it's very tightly bound, like very tight up to the proteins, it is not. And so there's a lot going on here, that's for sure. And so there are basically 3 different ways in which you can make these binding sites accessible by futzing around with the actual nucleosomes themselves, right? And so the first way is to essentially rotate those nucleosomes. So if you look back up here, right, this bit of DNA is wrapped around the nucleosome, and this spacing is intentional too. I mean, not intentional, right? We don't design our DNA, but it has been selected for. And so essentially there's a, every 200 base pairs, you'll find a nucleosome, right? Or so there's about from kind of about here underneath the eye and site to the start of the next nucleosome is about 200 base pairs, right? So these binding sites are spaced kind of typically in units of that. And so what we can do is we can simply rotate the nucleosomes, right? And what that will do is kind of move those nucleosomes along the DNA so that these binding sites are now between nucleosome core particles. Let's move this up. And this, as you imagine, requires energy, right? So ATP is the energy source for this process. And so this chromatin remodeling complex, CRC, is recruited. It's actually recruited by other modifications. And this is where it just gets kind of crazy. So other modifications to the histone and DNA actually recruits this, which then changes how accessible those binding sites are, right? So there's an awful lot going on here, and kind of one of the struggles I have teaching this is giving you enough detail so you can get a sense of that complexity, but without overwhelming you with detail, right? So it's a bit of a tricky balancing act. So when those nucleosomes are rotated, the DNA that was wrapped around them, it's like kind of like a rotating like a capstan with an anchor chain or something like that, right? When you rotate them around, now these binding sites are in the free DNA between those nucleosome core particles. And so now they're accessible. That's the deal. As they're accessible, now so even though the DNA can still be relatively tightly wound around these nucleosomes, the actual binding sites which are required for transcription are available. And so that then recruits all of the other proteins which are required to then recruit RNA polymerase and all of the proteins associated with that to begin transcription. And RNA polymerase kind of acts like a bit of a snowplow, or those plows they had on old-timey steam trains, push cattle off the tracks, right? They will push those nucleosomes off of the DNA as they go along, and they'll reassemble behind them. So actually being able to start the process is what is key. Another thing that you can do is kind of shuffle these nucleosomes along, right? So you can clear some space essentially, right? Simply by, it's like an abacus essentially. You just kind of push them to one side. Now you have bare DNA, which is accessible to transcription machinery. And on the similar kind of vein, I'll just kind of blow that out a little bit. You can just kick those histone proteins off entirely. And so this is a very dynamic process, right? Can't overemphasize that enough. This is something which is constantly opening up DNA for expression, shutting it down, moving nucleosomes around or rotating them, kicking them off, putting them back on all the time. It's an astonishingly complex, dynamic process. And so with the use of ATP, again, because this takes a lot of energy to do, you can actually just kick out nucleosomes, and now you generate that free, accessible DNA. So that's all about changing, in a sense, the physical structure or relationship of chromatin, right? Now we can get into some of the, there actually are somewhat subtle details. And this is all about histone modification and DNA modification. And of the two, histone modification is by far more complex, right? So we're going to spend a whole bunch of time on that. Okay, so we're going to do a little bit of recap work first. Question. What is the charge of DNA? Whoa, shit, that was quick. I didn't even have time to drink my coffee. Whew. All right, I got to ask you a more difficult question next time. Yeah, my brain isn't working that well right now. I'll save it for later. Okay, 100%. Second question, can you change the charge of DNA? Or actually, let's back that up a little bit. Why is DNA negatively charged? So Jonathan, Lindsay, you're both 100% correct. Why is DNA negatively charged? Now, think about the structure. Yeah, good job, Jonathan. Exactly. It's the phosphates in the phosphate sugar backbone. Okay, so do you think you can change that charge? if it's a fundamental part of DNA structure. Got a 50-50 chance. I guess that's a difficult enough question. Oh, let's just about to have some coffee. Correct, Lindsay. No, you cannot. Right. And that means that if you want to change how tightly histones are bound to DNA, what charge do histones have?
Positive.
Yeah, they're positive. Second question. What would you do? What would you do to histones to make DNA bind less tightly? So if histones are positively charged and DNA is negative, you can't change DNA. What are you going to do to the histones? What would you do to the charge, the positive charge of histones, right? So positive and negative obviously attract, right? And so if you don't want the negatively DNA to be attracted to histones as well, what would you do to the charge of the histones?
Make it negative as well.
Yeah, or neutral. Just neutralize it would work too. Don't think it changes its negative. No, it actually just neutralizes it, right? So you neutralize the positive charge of histones. And that is at the root of histone modifications. It's all about the charge of the histones and how tightly those histones bind DNA. So, oh, forgot. All right, I'm going to show you a quick picture. Okay, and so the part of the histone that is accessible, right? Because remember, these histones are kind of grouped together into an octamer, right? 4 pairs of proteins. And they have DNA wrapped around them. So the bit of the histones accessible are these tails, right? And these tails contain arginines and lysines. And if we go back, see if I can find the right, there we go, had it open from last time. Right, so all the way back to, what PowerPoint was that? 16, processing and translation, right? Hopefully this should look familiar. What charge do lysines and arginines have? Positive, right? And so if you want to neutralize the charge of the histones, you're going to be futzing with these amino acids. So I always kind of find this pretty neat because it really takes this basic properties of amino acids and turns it into a much bigger kind of deal, essentially. And so the way that we do that, I'm just kind of making sure I have my kind of thoughts in the right order, is by 2 opposing processes. We have acetylation and methylation, or acetylation. Methylation? Is that how it's pronounced? Well, you get what I'm talking about, hopefully, anyway. Yeah, nodding heads. Good stuff. So you've got 2 opposing processes. Methylation and acetylation. So by default, histones will bind DNA tightly equals transcriptional repression. Methylation, which is basically adding a CH3 group of lysines and arginines, reinforces that. And I couldn't tell you exactly how, because it's very complex and I don't fully understand it, to be honest. And it can kind of work in opposing ways too. It's just, yeah. Because as you can see, just to give you like a scary like look into the complexity of this, arginines, for example, they have 2 amino groups or amine groups. Each one of those hydrogens can be replaced by a methyl group, and the same for lysine. So for every lysine and arginine that you have in the tails, and there are a few, you can have mono, di, and trimethylated residues, all of which do different things. Monomethylated may be repression, dimethylated may be activation, trimethylated may be repression. It's just, to put it not politely, batshit crazy as far as I'm concerned. So I like kind of like surfing along the top of this topic, but diving too deep into it just gets stunningly confusing. However, acetylation is a lot more straightforward. So acetylation, missed out an L. And I think that's that group. I think, don't quote me on that, but I'm pretty sure that's right. Of lysines particularly, oh, sorry, I've got a dog lying on my foot.
It's kind of uncomfortable.
That removes the, oh, sorry. That removes the positive charge of the lysine equals loosely bound. Sorry, I can't see what I'm typing. Right. So generally speaking, and this is generally speaking, right? Methylation equals transcriptional repression. Acetylation equals transcriptional activation. And so if you look at it kind of closer, and these actually look a little bit like, don't know if anybody actually remembers Half-Life. It's a kind of cool game from many moons ago. These look a little bit like those kind of scary little aliens from that. But it's all about the tails, right? So when those tails are positively charged, they interact with the negatively charged DNA and they group it very, very tightly, right? So it's kind of really just glommed onto the nucleosome and not available for binding by DNA binding proteins. However, when those tails are acetylated, now that interaction is a lot weaker, right? So the DNA is still wound around those histone particles, right? Histone proteins, sorry. But now it's kind of a lot more loosely bound. And so there's actually enough DNA available that proteins can bind to it. And this is a very dynamic kind of state back and forth. So essentially you have, generally speaking, two kind of back and forth states. We have a methylated state, which is transcriptionally repressed. You remove those methyl groups and add acetyl groups, and now the DNA becomes more open, less tightly bound, right? So we go from heterochromatin to euchromatin. And now it's kind of available for transcription. And that's in a general sense, right? So acetylation is pretty reliably pro-transcription. Methylation is a lot more complex, right? It can be both. Anybody go on?
Right. Is there something that activates it that makes it become acetylated versus methylated?
Oh, absolutely. That was the next point I was going to get to. Thank you for reminding me. And so obviously there's a whole bunch of proteins that do this, right? And this is, so I'm going to tell you about this, and then I'm going to tell you how these all things interact, because it's, yeah, I'll do that first. Let's talk about the proteins first. And so basically you have, you know, you have two different opposing states, and you have two different groups of opposing proteins. Right, so pro-transcription will be things that add, generally speaking, things that remove methyl groups. So those are histone demethylases. Methylases. These are hard things to type out. And histone acetylases. These are called HATs. Can't remember off the top of my head the shortening for histone demethylases. I think they're just called DMs, but I don't want to tell you the wrong information. Right, so these are basically proteins that remove these methyl groups and add acetyl groups, right? So different proteins. And these are very, very specific for the different residues in these tails on different histones too, right? So there's a lot going on here. And so anti-transcription, in general, because I have to be careful not to make absolute statements here. These are going to be, what are they called? Histone methyltransferases, HMTs, and histone deacetylases. Acetylases, yeah. And those are called HDACs. So one group will kind of bias the histone, you know, charge essentially to being less attractive to DNA, right? So the DNA is loose. Those are things which add acetyl groups and remove methyl groups. When those acetyl groups are removed by histone deacetylases and methyl groups are added, then the DNA and the chromatin kind of gets more tightly bound and condensed, right? So, and as to what tells those to do it, well, we'll get onto that in a second, right? So they're kind of two opposing groups of proteins. And these are extremely specific to each histone, and crazy as it sounds, each lysine or arginine. More specific to the histones, not quite as specific for the residues. And so that generates what's called our histone code. So what was one I was looking at yesterday? So histone 3, and there are 2 of them, remember, because they're in pairs, is a very common one, which is, you know, studied. And so if you have, you see this particular code, what that means is lysine 9, which is the K9, on histone 3 is blank, 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 something, methylated or acetylated. And so you got all of these different residues, each of which has a potentially different outcome on transcription, or at least chromatin state. And those different residues can be modified in different ways, right? So you can see how utterly crazy this can get. Now, we talked about that already. Before we move on, has anybody got any questions so far? There's a lot of information to kind of absorb on this stuff. But what I want you to really get is that this is a kind of complex, dynamic state of how the DNA wrapped around these histone proteins is bound to those histones. Is it loose and accessible? Is it tight and inaccessible? It all comes back to accessibility, right? So it's really no different to whether or not those histones are present at all. I mean, it's a little subtler, obviously, because it's, or if the DNA is no longer on the histones, it's still all about how accessible is that DNA. All right, anybody got any questions? My foot is going dead. No? Okay. So that's histone modifications, right? And again, these histone modifications, because every time you replicate your DNA, you know, and if you think about DNA replication in terms of gamete formation, those histone modifications, those histones get reused, right? And so in each oocyte, there are going to be a bunch of histones, or each sperm, that have these epigenetic tags, right? This histone code, right? And so the offspring are inheriting not just the sequence, the information in the sequence of DNA, but essentially kind of a guide on how to read it. It's kind of the best way of thinking about it, right? How do we use that information, right? And that's in many ways what epigenetics is kind of all about. And so the next level is actually modification of DNA, right? So we're not changing the sequence of DNA. That stays the same. What we're changing is somewhat subtly the properties of DNA, right? How it looks. And if you remember, all of the proteins that we're talking about, like TBP, transcription activated proteins, transcription factors, and so on and so forth, right? The ones that bind DNA recognize a particular shape, right? The bases of DNA are part of that shape, right? And their individual charge and all those kinds of things. But they recognize like the whole picture, the topography, the sequence, all of that equals DNA binding. And so now we're going to change what that DNA looks like, right? And we do that by a process called methylation of cytosines. And as with histones, methylation of DNA is strongly associated with transcriptional repression. And so that's methylation is adding the CH3 group to cytosine. It's strongly associated. Oops, got my ass in the wrong place there. With transcriptional repression. And these Cs, right, and it does so in a couple of different ways, right? Essentially, it either changes the DNA, look, so that proteins that should bind to it no longer do so, right? And so transcription activated proteins, transcription factors, TBP, so on and so forth, right? And so therefore, those things don't bind, transcription doesn't occur. Or it changes that kind of sequence so that it's now recognized by repressors. So proteins that would not bind DNA normally, but when they see that change in kind of shape and look, now they'll bind. And these cytosines are found what's that called? They're called CPG islands. And the P is a phosphate, so it's basically a cytosine phosphate guanine. And those are found, oops, commonly in promoter regions. And they're called CPG islands. So you get a whole cluster of them, right? And it kind of makes sense, really, that they're in the promoters, right? Because that's where these proteins which activate or repress transcription bind DNA, right? And actually, I worked with a student once who did a project on this. So as kind of a little bit of context, right? What they find is that in certain cancer types, the promoters of tumor suppressor genes, that's what I was trying to think of yesterday. So cell cycle regulators, pro-apoptosis genes, various things which are basically work to prevent uncontrolled proliferation, cell division, right? The promoters of those genes are heavily methylated. Right, so we often think of cancer in terms of activating cell division, right, and promoting cell growth and differentiation, stuff like that. It's also about stopping the things that stop that, right? And so one of the ways in which that happens is by the heavy methylation of the promoters of tumor suppressor genes, which is really interesting, right? So it just kind of gets into how complex all of this is. And as with histones, we have kind of opposing proteins. So we have DNA, hang on, just trying to remember the correct term. What is it? So DNA demethylases. Those remove the methyl groups. and that's pro-transcription. And we have DNA methyltransferases. That can't be comfortable, dog. It's not comfortable for me. We should call DNMTs. Oh, sorry. Those add methyl groups equals anti-transcription, basically. Sorry, typing is not working well today. And the crazy thing is that all of these things, oh, here's a good, basically talking about methylation in DNA replication, right? So actually, you remember when we was talking about this in terms of DNA repair, so mismatch repair after DNA replication? This is the methylation which is used to identify the template strand, the old or parental strand, right? Because this methylation has to happen after DNA replication, right? So these kind of methyl groups are added to the newly synthesized DNA after DNA replication has finished. But the coolest thing, though, is all of this stuff talks to each other, right? So we have histone modifications, we have DNA modifications, right? And they talk to each other back and forth, right? And so let me just kind of bring up that. So quite often, and this kind of gets back, I think that was Kiana that asked the question earlier, like what makes those changes to histones? Well, those are essentially triggered by methylation, right? So where you have methylated DNA, what that will do is it will recruit other proteins. Those proteins will recruit, say, histone deacetylases, right? So we have a anti-transcription kind of marker that will then recruit ones which reinforce that in the histone proteins. Sorry, I don't know what it is with the weather at the moment. I've got really achy hands and terrible blocked sinuses. Don't know what's going on. Not a bunch of fun. Anyway, so that DNA methylation recruits the histone deacetylase. The histone deacetylase removes the acetyl group, right? So now we're getting even more anti-transcription, and that change will then kind of basically recruit a histone methyltransferase, right? And that then adds a methyl group to the histones, and in turn, that makes an even tighter kind of anti-transcription signal, right? So these things kind of reinforce each other. They kind of talk from one way back to the other. And you can see how this really shuts down transcription, right? It really does kind of strongly reinforce the kind of negative signal, and vice versa, right? It's not quite as strong going in the opposite direction, but it is, it does work that way too. Right, so another example here is that we have these kind of methyl groups here, right? Oops, I'm sorry, something's going on. Right, the DNMTs recruit the histone proteins. They will then methylate the lysines, and that further suppresses transcription. Right, so this is just an astonishingly complex process. And don't forget, this is also at potentially all of these different residues. We're not just talking about one or two, we're talking about lots of them. And we're also talking about multiple different levels of methylation, right? And so in many ways, right, we often think about like the complexity of gene expression being all about, control of transcription, like what proteins bind, turn genes on, repress, stuff like that. Before we even get there, we have just this astonishingly complex interaction between all of these different things to even make that DNA available, right? And so that's, in many ways, like before we even get to the complexity of everything else, right? It's just a mind-blowing, absolutely mind-blowing level of complexity. Okay, anybody have any questions before we move on? Because we're going to move on to transcription next.
I have a question about what's written on the slide we were just looking at. At
This
the
one?
bottom, the other one. I think it's 17.
Uh-huh.
On the bottom, it says both of these histone modifications reinforce gene silencing. I thought the first one loosened up the histones or the DNA so it could do gene expression.
Right, so that's what this acetyl group that was present promotes, right? So you're 100% at that. And so to shut down gene expression, not only do we need to take that acetyl group off, but if we add a methyl group, that even makes it even worse. Do you see what I mean? So it's kind of like degrees of, right? And so bare histones alone will bind DNA, but histones with methyl groups either bind DNA more tightly or prevent acetylation, essentially. Does that make sense?
Yeah.
Right. So so if you really want to shut things down, not only do you take off the marker which allows expression, but you also add another one which says no expression. Right. And so that's what's going on here, is that the DNA methylation is recruiting proteins which remove those acetyl groups, which even on its own will reduce DNA availability, right? Accessibility, really. But then that also then recruits another protein which adds a methyl group, and that shuts it down even further. Because now that lysine is not going to be available for acetylation, right? You'd have to remove it before you can add that acetyl group back. So it's like it's like having two locks on the door in a sense. Does that help?
Yeah, I just thought that it was that picture was just also further describing how they like loosened up so the proteins could bind to the TAP and the TBP things.
Oh yeah, absolutely. So really at the top, it's like this, right? So top of this slide, right, we have an acetyl group. DNA is loosely bound, right? And so if we want to reverse that, if we want to silence transcription or expression, not only do we need to remove that acetyl group, right, which basically turns it back to this form, right? But we also want to prevent this change from tightly bound to loosely bound happening again. Do you see what I mean?
Yeah, I understand now.
Yeah. And so methylation does that, right? It really locks down gene expression because it prevents the acetylation of those lysines. But yeah, good question. This is not trivial stuff to get. It's really quite complex. So good questions to ask. I appreciate it. Does anybody else have questions? Cool. All right, so kind of shifting on in gears and really kind of like now we're kind of going down at the individual level of individual genes, right? So chromatin modifications can affect like large areas of the chromosome. And, you know, we're really, now we're going down to the level of individual genes. And so if you remember back to what we were talking about ways away ago in terms of gene structure, right? Eukaryotic genes have, like prokaryotes do, promoters, but in eukaryotes, now we're going to split it into two parts. We've got our core promoter, right, which is all about RNA pol binding. And the core promoter is where the TBP binding site is, bidding binding site, and the basal transcription apparatus. That's a clunky way of calling it, but it's correct. So RNA pol and co assemble. Right, so that's the bare minimum, right, for a gene to be expressed. But as we've talked about before, that's not enough on its own, right? We also need a bunch of other proteins present to activate transcription, right? So simply assembling everything on the core promoter is not enough, right? And those other proteins, they bind to what's called the regulatory promoter. Oh, actually, before I get onto that, and it's always hard to define these things because it takes an awful lot of work to kind of narrow down like what bits of which sequence are involved. But generally speaking, the core promoter equals about, I don't know, 400 to 500 base pairs, give or take, right? It's not very big. That's the bare minimum. Then you have the regulatory promoter. Hang on a sec. I'm getting text messages from my daughter for some reason. And the regulatory promoter is where everything else binds, essentially. So you've got transcriptional activator proteins, repressors, silencers, etc. And this is very hard to define. And basically it can be, you know, anything from a few hundred base pairs to stretching, I don't know, millions of base pairs away. It's actually pretty bonkers, to be honest, you know, talking about this stuff. So, and again, all of these, again, what we're really talking about for all of this are DNA sequences, right? Specific sequences of DNA that are recognized by proteins. You know, and so if that DNA sequence is present and the protein is also present, that protein can bind, right? And so we have two different things kind of going on here, right? Do we have this particular sequence in the regulatory promoter of this gene? Pardon me. And if we do we have the protein that will recognize it, right? You have to have both present for transcription in that context to occur, right? And so this regulatory promoter is far kind of more nebulous, right? In kind of where it is, how big it is, what's there. But it's also where a lot of the complexity of the control of gene expression occurs, right? Because you can imagine if you have a particular DNA sequence that's recognized by one protein and one that's recognized by a different protein, now you have two different ways to turn on the expression of that gene or transcription of that gene, right? And so those little chunks of DNA are actually ways in which we think we gained complexity over kind of evolutionary time. And there's a really great example from butterflies, of all things. And so, you know how some butterflies have, you know, those spots called wing spots, makes them look like eyes of birds so that other birds don't go and eat them. Well, those wing spots are generated by the expression of a pigment that is in other, like moths and the like, is found in their eyes, or is expressed in their eyes, right? And so now somehow, right, it's hard to figure out exactly how, there was a little bit of DNA which said on in wings, right, that got added to the regulatory promoter of that eye pigment gene. And so not only do you have that gene turned on in the eyes, which is where it needs to be turned on, like seeing and stuff, but also it gets turned on in the wings. And so literally you get eye pigment expressed in the wings, right? So the gain of those bits of DNA, right? And they move around. Remember, we're talking about transposons, right? A transposon may carry that bit of DNA, or a virus may insert, which has, you know, that bit of DNA that completely changes the expression of that gene, where it's expressed in response to what, and so on. And so there are an awful lot of sequences that can be found in those regulatory promoters, right? And really, the core promoter has very little to do with complexity of gene expression. It's all about the regulatory promoter. That's where all those differences and gene-specific things are found in this regulatory promoter. So now you can see how hard it is to define and how big it can be, right? So do I have it? Oh yeah, I do. I'll just jump ahead a little bit just because it's fun to talk about. And so you see how there's this enhancer, right? It's a bit of DNA way elsewhere, not necessarily close to the gene itself, right? Which a transcriptional activated protein can bind to, and actually bends the DNA so that these now interact with the basal transcription apparatus. Well, in yeah, actually, let's just finish off with this because it's just fun. I mean, so I'm going to bounce back to the earlier parts where we talk about these kind of definitions in a little bit. I'll talk about those next Tuesday. I just want to talk about fun kind of fun thing to talk about, right? This is something I love talking about. So anyway, so this actually comes from teaching development. This is something that I cover in my Dev Bio course in a bit more detail than this, but this is still fun to talk about. And so essentially, as your you know, your limbs are developing, you have this limb bud form, and that limb bud then extends out, right, radially, to start laying down, you know, whatever bone that is, and muscles and so on and so forth, right? And at the end of that, you have kind of like a, I don't know if I can, it looks a bit like that, right? So you have this kind of bit of future hand, essentially, right? And as you know, pinky, thumb, they're very different from each other. And the reason they're different is because there's a protein expressed way down here at the bottom called sonic hedgehog. Told you that like fly people call things stupid names. Supposedly because the person that described it, his son was playing Sonic Hedgehog on Sega at the time. I don't know if that's a true story, but you know, it'll do. Anyway, so that one, that protein down here is basically pro-pinky, right? So if you have a lot of it, you get a pinky. If you have a little bit less, you get whatever that finger's called. And yeah, I have to be careful, right? Don't want to insult anybody. And then you get the I want to be rude to other people finger in the US, has a little bit less, right? And basically your thumb has no Sonic Hedgehog at all, right? And so the expression of both enhancers of Sonic Hedgehog expression, which are going to be expressed down here, and repressors of Sonic Hedgehog expression, which is expressed up here, those are found. Oh, dog's having a nightmare. Way further away from the actual gene itself, right? Now, if you mutate those, right, so you essentially now you have Sonic Hedgehog expression up here, and if you stain for it, it looks something like this, right? You get this. This is that's probably looking at mRNA, I would guess. So that's probably like a northern blot. And these are hundreds of thousands of base pairs upstream, right? A long, long way. You get extra toes. Because basically you're now saying pinkies, or I don't know what the equivalent is in cats. I don't know if they have like digit stuff. But anyway, cats don't have thumbs, thankfully, otherwise they would rule the world, right? But if you have extra expression of Sonic Hedgehog up here, instead of turning your thumb into a pinky, now you get essentially, let's see if I can do it right, something that looks like that. This is called polydactyly. So you get extra digits. You get this in people too, right? But it's particularly common in cats. Shoot, what was that guy's name? Can't remember. Oh, Hemingway, that's it. Ernest Hemingway had an island somewhere, and supposedly there are lots of Hemingway cats that have extra toes. And so depending on how much you change that, which again, going all the way back up here, depends on the mutation that affected this enhancer or oppressor, basically determines how many extra toes you have. And this was actually from a student of mine, sent me this picture of her cat. So when I talked about this, she's like, ah, my cat has extra toes. And this is Mr. Tuxedo. And as you can see, if you look in the picture in the background, he's not a very happy cat. So, and I think the Guinness Book of World Records for number of toes on a cat is 32. Right, they should have 20, right? I think, or maybe even less than that. Kind of depends on how you count them, right? So that's a lot of toes. And that simply is not a change to the actual coding protein itself, right? It's not a change to the core promoter. It's not even a change to the regulatory promoter that's right next to the gene. It's a change that's way, way, way away, right? That changes the expression of that gene. It's super, super cool. Anyway, that's more than enough talking from me today. I'm going to love you and leave you. Hope you have a lovely weekend. And I'll see you soon.